This is your instructor Kwame Shen, and we are continuing on the second part of the lecture Chemistry of Life. So in this part 2, what we are going to concentrate on are the biological molecules that exist in living systems. We'll look at the inorganic and the organic compounds that exist in living organisms. In the inorganic compounds, which we typically describe as compounds that lack carbon, and we're talking about water, which is the most abundant molecule, oxygen, carbon dioxide, which all play very important roles in our survival on it. Water is essential for life on Earth, and that is evidenced by the percentage of the Earth's surface that is covered by water, about 70% also. They also an important component in terms of how our cells are able to absorb the nutrient that it needs. So these nutrients would have to dissolve in water before the cells or living organisms are able to absorb them into the cells and use them for their cellular activities. And because of this property, water is a very essential medium that without it, many chemical reactions would not occur and life as we know it wouldn't exist. Cells have about 70% or so of their constitution being water. So why is water this unique and why is it this abundant? And the reason comes down to its polarity. Water is a polar molecule. Water is a polar molecule because water, which is made up of oxygen, one oxygen atom and two hydrogen atoms, has a slight negative charge on the oxygen and a slight positive charge on the hydrogen. If we look at the periodic table once again, what we notice is that as you move across periods from group 1 all the way to the last group, what you notice is that you're moving across a property of atoms known as electronegativity increases. Electronegativity is a measure of the ability of an atom to draw another electron from another atom onto itself. So hydrogen is at this far left end, oxygen is at the far right, and as you're going towards the right, electronegativity increases. So this property that oxygen has to draw more electrons onto itself than hydrogen has makes oxygen more negative. So it's slightly negative because more of the electrons, which is negatively charged, are towards it, and hydrogen becomes slightly positive. So this creates a polarity. The molecules of water carry these charges, these slight negative and positive charges around, which makes them polar. And that accounts for many of the important properties that water possesses. The polarity makes water a very important buffer. A buffer is a substance or a molecule that maintains the pH irrespective of changes that occurs within the system. So even if there are slight changes here and there, it is still able to maintain the concentration in terms of if you're talking about hydrogen ion concentration, if you're talking about temperature. So in this particular instance, we'll just stick to the temperature aspect of the buffer. So it makes water such an important buffer in terms of temperature regulation. And the reason is that for each atom or each molecule of water, there are 4 hydrogen bonds that are connected to it, to its neighboring water molecules. So as you have a lot of water molecules here, each water molecule is connected to 4 hydrogen bonds. That makes it very difficult to break all these bonds, and so water has a high boiling point, which is very important, and so that organisms that live in water, even when you have very high temperatures. It takes a while for the water temperature to get to the point where um, it's too much for these organisms to bear. It's the same for us as humans. We have another living organisms who have water being an important component of their blood systems, and that helps make it a good buffer, so that when there are slight changes in temperature, we are still able to survive. If you take an equal volume of water and ice, what you will notice is that there are more water molecules in the liquid states of water than in the ice states of water. And the reason is that, excuse me, the bonds in water becomes crystallized. So becomes crystallized and well laid out as you have in here once water becomes ice. And once it takes on this state, there are fewer molecules that can be packed in there as compared to the liquid molecule. The good aspect of that is that because if you compare the two, you have more molecules in the liquid water than the ice water. The ice water then becomes less dense, and so it floats on top of the liquid water. This is perfect for maintaining aquatic life and for sustaining the lives of organisms that live in water. Because when the temperatures drop in an environment and you have ice forming, the ice will just sit on the top of the water because it's less dense, and you still have water available in the liquid state for the organisms below to continue to live on. That's which is very crucial for life on it. Water's polarity also makes it an excellent solvent. You have many polar substances that are able to dissolve in water. So if you have, for instance, sodium chloride, which is a polar molecule, remember it has ionic bonds. And with these bonds in the presence of water, it's very easy for them to dissociate, to dissolve, so that the sodium will be drawn more towards the slightly negative oxygen in a water molecule, and the chloride will also be drawn more towards the slightly positive hydrogen atoms in the water molecule. So it makes it a very good solvent. The polarity also makes it easier for water to split up molecules, which we call hydrolysis, or for water to be added onto molecules and help to split them up. And many chemical reactions take place in this manner. The amount of hydrogen ion in a system also confers a certain characteristic on it. If you have more hydrogen ions produced by a particular molecule in the presence of water, then that molecule is described as an acidic molecule. Molecules that have more hydroxide ions released when mixed with water are bases. And so if you look at HCl, dissolve HCl in water, it will dissociate into hydrogen ion plus and chloride ions. The presence of the hydrogen ions when HCl dissociates makes HCl an acid. If you take sodium hydroxide, it will dissociate into OH minus and Na plus, and the presence of the OH minus makes sodium hydroxide a base. However, when it comes to salts, once in solution, they dissociate into ions, but these ions are all ions except ions of hydroxide and chloride. So you would have salts that will dissolve in solution, but you cannot have these ions being hydrogen or hydroxide, because once you have that, then it's either an acid or a base. And typically you would have an acid and a base coming together to form salts and water. Okay. So acids and bases has led us to create what we call the pH scale. The pH scale, this is an example of a pH scale running all the way from 0 to 14. 1 to 14, however you put it. But it is a measure of how much hydrogen ions you have in a solution. If you put a molecule or a compound into water and it creates a solution, how much of it is hydrogen ions and how much of it isn't hydrogen ions. So if you have more hydrogen ions than hydroxide ions, then that's an acidic solution. If you have the equal amount of hydroxide ions and hydrogen ions, that's a neutral solution. If you have more hydroxide ions than hydrogen ions, that's a basic solution. And you have molecules and compounds that exist, including food and wine that we consume, that have different, the ones that are consumable, some are not, that have different pHs. So it will represent how acidic or basic this solution is based on the charge, ranges from 1 to 14. 7 is neutral, less than 7 acidic, more than 7 is basic. The interesting thing is that for most biological systems, the pH that exists within living organisms is around 7, which is neutral. If you look at the pH here for water is 7. Look at the pH of human blood, just around 7.45 thereabout, which is close to 7, which is the perfect neutral pH. And pH, because of pH, we are aware of the buffer property that certain molecules have. I've already alluded to the fact that water is an important temperature buffer, so that it doesn't fluctuate so easily, and that you need a lot more heat to raise the temperature of water. And we employ this principle when we are growing bacteria, we're cultivating bacteria in the lab. For instance, we take a compound potassium phosphate and mix it with the growth media that we use in culturing bacteria. And the reason being that when we provide the bacteria with the nutrients that they need in order to flourish, because we want to see them grow, they produce byproducts that are harmful to them because of how acidic or basic these compounds are. So we add buffer to growth media, and so that even when they metabolize these food compounds and they produce the acidic or basic end products, the buffer compound that is part of the media will still keep the pH within rates that is good for them to continue to grow and survive in the media. We look at organisms like Escherichia coli. They all have has a minimum and a maximum range in terms of the growth temperature, the growth pH. So that without the buffer, it is possible that E. coli can release acidic products that will end up moving the pH beyond or below the 4.4 and causing it to actually die. So our knowledge of buffer and the pH ranges of these microorganisms help us to cultivate them in the lab and to study them. For the organic compounds that we have in living organisms, we call them organic because they always contain carbon and hydrogen. Carbon has 4 electrons, and so it has a valence of 4. It's able to form several different bonds, different combinations, to form rings, different chains in terms of the lengths, and that makes carbon very versatile. So we see carbon forming the basis of many compounds in living cells. It's all over us. So in addition to the various shapes that it can assume, you have other compounds, organic compounds that are also part of the compounds that carbon forms of hydrogen, oxygen, sulfur, phosphorus, and nitrogen. So typically you would have a chain, a carbon chain, but a carbon chain would have what we call a functional group that is attached to that skeleton. So this is the carbon skeleton. You have a functional group attached. A functional group, you're talking about groups of atoms that are attached to the skeleton. So for instance, in this example, you have SH that is attached to the carbon chain. You have two carbon chains. And this is an example of a functional group. You also have OH that is attached to it. And that is also a functional group. Functional groups are essential in that they confer on molecules and compounds specific characteristics. And so it makes them behave in a certain way in reactions. The functional groups would always be attached to a skeletal carbon, so they don't exist on their own. And organic molecules may have more than one functional groups, like the example that we just looked at. So there's an example of an organic molecule, beta mercaptoethanol. It has both the OH functional group and the SH functional group. We can also use the functional groups to group compounds because then we know they all belong to the same family. They all behave similarly when in reactions. So for instance, you have the OH functional group, and that belongs to alcohol. So alcohol would have the OH functional group. Amine has an NH2. Alkene has a double bond between carbons. Alkyne has a triple bond between the carbons. Carboxylic acid would have a C double bond O and an OH as well. And so we're able to use this to actually group the different organic compounds that are in living organisms. Amino acid is a very important organic compound. It's an example of a compound that has more than one functional group. So amino acids would have an amino group. It would also have a carboxyl group. But in addition to that, it will have a variant. So for the amino group and the carboxyl group, it's the same for all amino acids. However, the variant always changes. So for glycine, it's an H that's attached here to this carbon. For tyrosine, you have the ringed alcohol that is attached. And then for cysteine, you have CH2SH that is attached to the carbon, the central carbon. So organic molecules may have several functional groups attached to it. Lipids and proteins have carboxyl group present in them. Proteins alone have amino groups as a functional group that exists in them. Lipids and carbohydrates have a hydroxyl group as a functional group that exists in them. And ATP and DNA has phosphate functional groups as parts of their composition. Molecules that we classify as organic molecules may also combine to form very large molecules, which we call macromolecules. And examples of these macromolecules are carbohydrates, lipids, proteins, nucleic acids, and the like. For macromolecules to exist, there has to be several smaller molecules called monomers. So these monomers have to combine by covalent bonding. So you have repeated covalent bonding. So if this is a monomer, it binds with this other monomer, and that also binds with this, and it forms a chain. And you have covalent bonds that are bind them together repeatedly. Now, when you have monomers coming together to form these bonds in a chain fashion like that, that results in what we call a polymer. Several monomers coming together is a polymer. The process is governed by dehydration synthesis, so it's a synthesis reaction. You have glucose and fructose coming together. Water is removed. That's the process of dehydration. But there is a synthesis, a new molecule is produced. Similarly, water can be used to actually break down molecules. So you have maltose, that is two glucose molecules that are joined together. If you add water to it, that's a process of hydrolysis, and that can result in the dissolution of the bond between the two glucose molecules. You need to note that for dehydration synthesis reactions, energy is used. Note that new bonds are formed, so energy is used in these reactions. For hydrolysis reactions, bonds are broken, so energy is released. For the macromolecules, we'll first look at carbohydrates. Carbohydrates are made up of monomers. The monomers may be either monosaccharides or disaccharides. A monosaccharide is the simplest unit of carbohydrates that exists. A disaccharide is when you have two monosaccharides coming together. Examples of carbohydrate monosaccharides are glucose, fructose, and galactose. For the disaccharides, you have sucrose. When you have glucose and fructose coming together, you have lactose and maltose as well. Carbohydrates, because they have monomers, can form polymers. So there are polymers that exist, and these are called polysaccharides. So you have monosaccharides, you have disaccharides, you have polysaccharides. They are a bit more complex. Examples are starch and cellulose. You have starch, that's one way that food is stored in plants. You also have cellulose, which is also another way that polysaccharides are packaged in plants as well. It comes down to how the bonds are formed between the monomers, which are glucose molecules. And so for starch molecules, you have all the OH on just one side. The linkage in terms of the bonding is called 1,4. This is 1, carbon 1, this is carbon 4. So that's 1,4 of the alpha glucose monomers. And then you have 1,4 linkage of the beta glucose monomers. What you will notice is that for alternating monomers in the cellulose, the OH is on the opposite side. Okay, so there's on this side and this is on the other side. Whilst in starch, they are on the same side. Carbohydrates are an important source of energy for a lot of living organisms, including microbes. And so there are tests that you can do on microorganisms to see whether they're able to ferment carbohydrates. Are they able to use it as a source of energy or not? And so starch is one way that plants store carbohydrates. And in terms of need, they rely on the starch as a source of energy. It's similar in animals. We store carbohydrates as glycogen. Plants structurally also use cellulose. It's a cellulose in the cell walls of plants that gives it that turgidity, that makes it turgid, and that it's able to, the cells are able to be filled up with water, and plants can stand tall as we see them. Nucleic acids exist also because of carbohydrates. A component of the nucleic acid is a sugar, the ribose sugar, or the deoxyribose sugar, depending on which nucleic acid that you're talking about. Then you have lipids, and lipids or fats. So you have a molecule of fat. A molecule of fats is made up of glycerol with either 1, 2, or 3 fatty acid molecules combined. So you'll have 1 glycerol in this case. This is glycerol. It's called glycerol. You have 3 carbons that are bonded to each other, single bonds, and each of them is attached to a hydroxyl group. Each of them is attached to a hydroxyl group, and then they are tied to one fatty acid molecule. And so this is a monoglyceride. This is an example of a monoglyceride. So one glycerol and one fatty acid. And this is fat. It's a fat molecule. It is called palmitic acid. You could also have two or more. So you could have a triglyceride where you have three fatty acids that are attached to one glycerol molecule. Lipids also play an essential role in helping a lot of organisms to actually survive harsh conditions. And so if we look at the Arctics, you have polar bears and all these living organisms that thrive in these environments, and mainly has to do with the fact that they're able to insulate themselves from the changes in temperature that happens in the environment, and also use some of these lipids as energy storage that they rely on the time when they hibernate and as a source of their energy. But more importantly, the membranes that exist in living organisms are made up of lipids, and they are essential. They are essential for the survival of these organisms. Mycobacterium tuberculosis is a very pathogenic bacteria that causes tuberculosis, and it's mainly because of the rich lipid contents that exists in its cell walls. And so we have to use actually a different test, acid-fast test, in order to actually figure out whether it is that bacteria that we are dealing with or not. It makes it more pathogenic because of the lipid content there. Now, for the membranes of living organisms, you have lipids that are attached to phosphate groups. So you have a phosphate group here attached to lipids. So you have glycerol and you have fatty acids. This is saturated, so there's no double bond. There's unsaturated. You see a double bond here. Now, in reactions where you have cells, when water is present, the cell membrane assumes this arrangement, where the tails are where you have the fatty acids. The tails are hydrophobic. Hydrophobic is a fear for water, and the heads where the phosphates are hydrophilic, so they love water. So they arrange themselves this way, and this arrangement is very important in moving molecules across the membranes. It's also important in maintaining the integrity of the membrane, and many other important characteristics that it confers on a cell membrane. Then you have steroids. Steroids are made up of fused rings of lipids. So steroids typically would have 4 fused rings, 1, 2, 3, 4, that are fused together. It is important. It's also an important component of the plasma membranes of animals, especially cholesterol. It prevents the packing of fatty acid chains and the hardening of the membrane. So the membrane is very fluid, and the fluidity of it is mainly due to the unsaturated fatty acids that are in there. Having the right combination gives the cell membrane the right integrity that it needs to maintain cellular activities. Then you have proteins. Proteins are so essential because we are working chemical reactions, and the same with other living organisms. And chemical reactions would only proceed when enzymes are present, are produced. Or chemical reactions will equally be controlled by chemicals as well. We have DNA replication, which is also controlled by or governed by proteins. Transportation, you have transport proteins that are involved, responding to stimuli, the movement of impulses from one part of the body to other. So proteins are so crucial. They are polymers proteins are polymers that are made up of monomers called amino acids. Amino acids are the building blocks of proteins. So they are the monomers that make up. And they have an amino group, a carboxylic group, and a variable chain. There are 20 naturally occurring amino acids that occur in biological systems. And they all will have a variable chain. So it's the same. They have the amino group, they have carboxylic group, but it's the variable chain that differs. And so it confers on them a different characteristic, because that's a different functional group that you have there. Amino acids join together to form a peptide bond by the process of dehydration synthesis. So water is removed, you have the OH and the H that are removed, and then you have the NH and the C double bond O coming together to form a peptide bond. So this is an example of a dipeptide. You could have several of them coming together to form a polypeptide. And how a protein is structured, how it's folded, is essential for its function. So if how the protein is folded is changed, it affects how the protein functions. So you have a primary protein based on the fact that it's just a single peptide, just arranged in chain. So there is nothing fanciful about it. That's a primary. Then you have a secondary proteins. Secondary proteins are folded and more like a helix. And so that's a secondary protein. And then you have tertiary proteins that are folded more into a 3D structure. And so the structure is important to its function. There are certain conditions, such as high temperature, such as acidic conditions, or highly basic conditions, or very salty conditions, that could cause proteins, the structure of the protein, to change. We call that process denaturation. If a protein becomes denatured, it cannot carry out its biological activity. It cannot catalyze a reaction or perform the role that it's supposed to perform. To revert it back to its original folding, it's impossible. It's almost impossible. So when protein becomes denatured, it most likely would result in some loss of some functioning at the cellular level that may present in the form of some symptoms that we exhibit as living organisms. Some of them may be benign, so even though the protein has been denatured, it might not really result in any debilitating effect in the living organism. Then you have nucleic acids. Nucleic acids are also very large molecules. They are polymers, so they are made up of monomers called nucleotides. Examples are DNA and RNA. A nucleotide, which is the basic units of a nucleic acid, is made up of three important components. It's made up of a phosphate group, a 5-carbon sugar, 5 carbons, so 1, 2, 3, 4, and 5. A 5-carbon sugar and a nitrogenous base. So this is a nitrogenous base. So this is a component of a nucleotide. DNA has 4 types of nitrogenous bases, and so I'm referring to this base here, which is part of a nucleotide, the nitrogenous base. DNA has 4 types of them, 2 groups. The first group are called pyrimidine. They have only 1 ring of carbon. Then you have the purines, which have 2 rings. The pyrimidines are thymine and cytosine. The purines are guanine and adenine. It's the same in RNA, except that in RNA, thymine is replaced by uracil. So we say in RNA, U replaces T In RNA, U replaces T. Nucleic acid nitrogenous bases will only pair up in a certain manner. They will only bond with each other in a certain manner. And because they only bond in a certain manner, we refer to that as the complementary pairing. They complement each other. So if there's no, it's like a lock and key. It's not a right fit, it's not going to form a bond with it. Now, thymine would only bond with adenine. So you would have thymine bonding only with adenine. You would have guanine only bonding with cytosine. You see 3 hydrogen bonds here, you see 2 hydrogen bonds here. So you actually need more energy to break up a bond between cytosine and guanine than you would need to break up thymine and uracil, thymine or uracil and adenine. Nucleic acid also form bonds. So you have these, they are formed by bonds between the monomers, the nucleotides. This is also an example of dehydration synthesis, where water is removed in the formation of the bond. The bond is formed between the phosphates group of 1 nucleotide and the 3rd carbon of the neighboring nucleotide. And it's an example of a phosphodiester bond. That's what we call it. DNA is arranged it has 2 ladders, so DNA has 2 strands, and they are each connected by their bases. So you have hydrogen bonds that connect these together. You have phosphodiester bonds that connect the ladder together. And then it's coiled together in a helix form. RNA has only 1 strand. The nitrogenous bases are the same, except that thymine is replaced with uracil. RNA is important for protein synthesis, so we have different types of RNA that are all involved in protein synthesis. Then, and a very important nucleotide is ATP. So this is a nucleotide. Technically, it is an adenine nucleotide. However, this nucleotide has extra 2 phosphates attached to the phosphate group that is part of the nucleotide. So you have adenine, you have a ribose, a sugar, and instead of just having 1 phosphate attached, you have extra 2. A lot of energy is in this particular bond, the 2nd and the 3rd phosphate group, and that's the energy that is released to fuel cellular activities. So this brings us to the end of the Chemistry of Life lecture, and we've gone over starting with atoms, moving to molecules and compounds, looking at the different chemical reactions that take place, the different bonds that take place, and then we've come on and looked at inorganic and organic molecules and the different properties and the advantages that they confer on living organisms.